All right. Good afternoon. Thank you all for attending. My name is Megan Troy. I'm an Assistant Director of Graduate Admissions here at Halmos College at Nova Southeastern University. And today's virtual discussion is about our brand new program that is starting this fall, our Master's in Science in Biological Sciences with a concentration in bioinformatics. Today, we have the faculty from the Department of Biological Sciences here to talk with you and an alumni of our master's program. And we're hoping if you have questions, please feel free to put them in the chat. And in the later part of the discussion, I'll open up the floor where you can vocally ask your questions as well. So first, I'd like to introduce you guys to the department chair. We have Dr. Emily Schmidt-Lavin, and then she will take over to introduce herself and her faculty, and then we'll get started on the topics that we have.
Well, hello, everyone. Thanks for coming today. We're excited to talk to you about our Masters of Biological Sciences, and in particular, the Bioinformatics concentration. So I'll just get us moving along so we can especially get to our faculty and particularly our alumni who's visiting with us today. So Dr. Johnston's next.
Hi, everyone. Glad you all could make it. So I am Matt Johnston. I'm a faculty member here in the biology department. I help to manage the new bioinformatics concentration as well as the thesis and capstone. So my background is in computational biology, not necessarily genomics, but data is data, as I say. So I'm kind of going to be your point of contact for any questions about the program. I also run an orientation right at the start of the your first semester, so you can feel free to ask me any questions during that time as well. We do have a couple more faculty members here, 3 more faculty members that teach in the program. I also teach one of the courses, so I'll let them introduce themselves, Dr. Gill, Dr. Lopez, and Dr. Ozga. So go ahead, Dr. Gill.
Good afternoon, everyone. My name is Navi Gill, and I'm assistant professor in the Department of Biological Sciences. My expertise is in the field of genomics and bioinformatics, and I use the tools and techniques in bioinformatics to answer biological questions. And I will be teaching a couple courses in this concentration, so I'm excited to have you guys on here to learn more about what we are going to offer you in this course. On to you, Dr. Lopez.
Thanks, Dr. Gill. Yeah, this is Joe Lopez, and my background is in molecular biology and genomics, evolutionary biology, and phylogenetics. And I've been applying much of this to my work since 2007 at Nova, mostly to marine invertebrates and microorganisms, because everything has DNA, and this is one way we can understand their evolution, comparative genomics, and we have to start learning how to understand and work with the data. So that's kind of one of the goals we have in training this next generation of students, biologists, to use the data and work with it, because these are usually huge data sets, complicated, require a bit of computational know-how, some programming, but it's not essential, but just being comfortable with it. So we'll hear a lot more about that, and we have the faculty to help out. So Dr. Ozga, next.
Thanks, Joe, and thanks, Joe. Thanks, everybody. I'm happy to be here. Yeah, I just finished up my 4th year here at Nova. I'm an assistant professor. I'm actually a classically trained molecular anthropologist. So my interest in the bioinformatics field has mainly been from a primate perspective. So some of the work I've done in the past has been with dealing with ancient genomes, DNA recovered from a variety of sources from very long ago up to 10,000, 20,000 years ago, that require a whole other set of bioinformatics kind of skills and techniques to make sure that you're not getting contamination from modern sources. I work with some folks that deal with non-human primates like gorillas and chimpanzees, and I've been interested in essentially how the oral microbiota has evolved in different human populations and then moving from great apes into humans. And currently I am also involved with the College of Dental Medicine. I'm doing some projects related to how different oral microbes are, basically what types of bacteria are present in the mouths of healthy humans in South Florida. There's a really diverse community of individuals down here that kind of allow us to ask some unique questions. And I am teaching bioinformatics, the introduction of bioinformatics in this graduate program that just kind of covers some of the basics of a lot of the techniques and tools and data formats that we use. And then we're starting up a new course that I'm teaching in the spring for the first time, which is going to be devoted to metagenomics. So dealing with some of the foundational components of that field of genomics and the techniques associated with it. So welcome to everybody, and I'm happy to be here.
Thank you all. So this is just an idea of some of the areas that our faculty are focusing on. So students, if you have questions about any of these areas in particular, this is always a good thing to kind of jot down. If you're interested, then maybe you might find a faculty member that's also interested. But I'm going to turn it back over to the faculty so they can tell you a little bit more about how the bioinformatics program itself will work and some of the courses that are going to be involved.
Yeah, so this is our flyer, and you should all have received this already, or you will be receiving this from Megan. So this lays out the coursework that you will take during your studies. So this is a lockstep program, which means that you take specific courses during specific semesters. It's 16 months long, starting in the fall, then winter, you'll have the summer off, and then you'll graduate the following fall. So first semester is 4 courses, second semester is 3, and then the fall of your second year is an additional 3 courses. So the first semester has some more introductory courses. One of the courses is biological data science that I teach, and that kind of introduces you to large data sets and big data and a little bit of programming as well. So skills that you will be using along the way throughout your journey. So in the semester 3, you do have an opportunity to take 1 elective. So there's a list here of courses that you can choose from. If you have a different course that you think may fit into your interests, we can also discuss you taking that. So again, you should have received this, and if you have not, just let us know. And this basically lays it all out on the line for you, gives you a little description about each course, and gives you a good idea of what is involved in the course work. Again, all these courses are online as well. We do have some on-ground options if you're interested in that, but we can discuss that at a further time. So the concentration, again, is 16 months long, so under 2 years. And basically, we're going to be dealing with genomics data. Now, that doesn't mean you're, as I said previously, data is data. So I don't work specifically with genomics data, but I work with other large data sets. And so by going through this concentration, you have the opportunity not only to do genetic work, but you can do other kind of data mining work if you so choose later on. But again, primarily, we're going to be focused on genomics data as well. Fully online again, so you don't have to be on campus. You can be dispersed anywhere to take the courses. Some of them are taught hybrid, like the course I teach is a hybrid course where I actually do live lectures. Other courses are going to be completely fully online without live lectures. Okay, you want to move to the next slide there? And I will let, I believe Dr. Lopez, Yvonne was one of Dr. Lopez's students, so maybe he can do a brief introduction here to Yvonne. Yvonne is one of our alumni.
Yes, thanks, Matthew. So Yvonne Desplat graduated from our department in 2021. He worked in my laboratory at the Guy Harvey Oceanographic Center, and carried out a very interesting experiment on or analysis and a set of experiments on marine sponges that were exposed to the Deepwater Horizon oil spill or oil. So we actually carried out these experiments in a laboratory, controlled, and exposed them to oil doses, and then looked at the gene transcription of the response of the sponges. So And then he's carried on after that. That included a lot of transcriptional RNA data, RNA-seq data that he had to work with. So he's now in California at a biotech company called Jump Codes, and he's been there for the last couple of years in San Diego. Yvonne, you want to tell us more?
Yeah, thanks, Joe, for the introduction, and hello everyone who's attending this meeting. As Dr. Lopez said, I was part of his lab for my master's degree, and I'm not going to go over what you just explained, but after I graduated and basically during my thesis, I had to take a few classes that were that actually I'm sorry if I forgot your name. Dr. Ozga will teach. I took Intro to Bioinformatics, even though that was a different teacher, but and then I moved on to learning a few other tools from other thesis advisors that were all useful because, as Dr. Lopez mentioned, the project that I took up on was heavy on bioinformatics analysis. And so it required me to learn quite a few tricks and tools and how to use those programs and softwares to analyze the data, because yes, you generate data in the lab, but how do you make the data speak and interpret it to really get the meaning of your experiment is what is key. And so all the classes that you will take during that program are actually going to help you not only, as Dr. Osga said, in bioinformatics, but programming is huge nowadays. Like the number of tech companies itself that are using bioinformatics software or programming software or programming language as a thing itself is just huge. And what I learned during my thesis under the supervision of Dr. Lopez is some skills, that I used every single day here. I'm mostly a wet lab person, so meaning I do the experiments in the lab with the pipettes and the reagents and that sort of stuff. But I'm also working on interpreting the data that comes off the sequencers and using those softwares. And so the stuff that I learned during my thesis is really applicable right now. And obviously you learn a good chunk of skills and how to use these in the program that you will be attending. But after that, it's going to be a constant progression. So and once you know how to code, you can do basically anything, because as Dr. Osga said, and everybody will tell you, the data sets are getting bigger and bigger and bigger every day. And so knowing how to skim through the data and make it speak is really important. But yeah, basically based off in California right now, called a company called Gemcode Genomics. We're actually very heavily relying on metatranscriptomic and metagenomics data that you will probably see throughout the course. And nowadays, if you take a look at a lot of the fields are heavily focused on metagenomics and metatranscriptomics, because we are understanding that everything is related and it's not a single aspect study. And so there's different impact of different microbes or different genes. And as a whole, if you just study one thing, you will not get the full picture. So yeah, that's a little bit about me and what I've been doing here. So I don't know what's the next slide about, but if you have any questions, feel free to reach out. And I think Dr. Schmidt-Lanvin and Dr. Lopez have my email, so you guys can email me.
Thank you so much. I'm going to quickly just go over admissions. Then I'm going to turn it back over to the faculty so they can talk to you guys more about the opportunities that are involved in the bioinformatics program. So for any student that's interested, the program does start this fall. The application deadline for fall is going to be July 15th. As Dr. Johnson mentioned, it's a lockstep program. So the program begins every fall semester, and there's 3 terms that you guys will be involved in. That may change in the future, depending on how the program runs, but just letting you know, if by July 15th, you have your application in, these are the documents that need to come with it. We need all official transcripts. So any institution that you've attended, you must send in your official transcript. If you're an international student and you're interested in attending, you will need to have your transcript evaluated. And of course, feel free to reach out to me and I'll tell you the services that you can use to get that transcript evaluated. 2 letters of recommendation. We ask that at least one of them comes from a former professor. So someone that you've taken classes with, ask them to go ahead and write that. In your application, you will provide their email and their name, and we'll send out a form for them to fill out and to attach their recommendation letter for you. A personal statement that's going to be written right into the application. That's just your way of telling us why you're interested in this program, why you think this program might be a good fit for you. Generally, we're looking for students that have a 3.0 GPA or higher, but we will consider students whose GPA fall a little bit under that. Of course, we're looking at your full package. The GRE is waived for this department, so we are not requiring students to submit their GRE scores. If you have test scores available and you would like them to be a part of your application process, so you did really well when you took the GRE, you're more than welcome to submit that. When I put your application package together, I'll include your GRE scores if they're available. Quickly, just to go over the next slide, I think that's always the important thing for students is how much does this program cost? So because this program is situated as a fully online program, and again, if students have an interest in doing some of their coursework on campus, you do have to talk to us, but generally this program is fully online. So the fees for the program tend to be a little different for students that are applying versus a student who's going to be fully on campus for a program. But the tuition is a per credit cost, and it's the same for all of our biological sciences programs. So it's $14.44 per credit hour, and this program itself is 30 academic credits. Again, you are doing this program over 3 academic semesters, so you can kind of do your math in that sense. Okay, so I am going to turn it over. Dr. Lopez wanted to share some slides with you guys as well to talk a little bit more about the program. So I'm going to stop my share and then allow him to go ahead and share his. But again, if you have questions, feel free to put them into the chat and we will go ahead and answer. Go ahead, Dr. Lopez, you should be able to share.
Okay. I didn't really have any slides, but I just wanted to mention the context again of genomics and bioinformatics, that
Okay.
you can apply, these skills that you learn across the board, because again, everything has DNA, and everything that we know of that's alive on this planet has cells. So we're part of a consortium called the Earth Biogenome Project. And several of us are involved in that. And this is trying to sequence all of life very ambitiously. But the start of it is trying to get whole genomes of as many species as possible. And all the species we know of are 1.8 million in total. These are just the eukaryotes. And this consortium is starting taxonomically. Let's see if we can get the families together, then the genera, about 180,000 genera as such. But this just shows the cavalcade of much of the data sets that might be available, in addition to the human genome project, right? So you would be able to apply that type of skills to the human genome project. And just as an example of what's going to be involved when generating these data sets, we're part of, this is going to require a village, as you might have heard, to carry out these large projects. And these are some of the affiliated networks within the umbrella of the Earth Biogenome Project. That's both taxonomic as well as biogeographic consortia within specific areas like Africa, Europe. These are all just going to be very interesting areas to look at and study. So your skills in this curricula, we hope, can show you the way and train you in some of those aspects. And we'll be talking about that if you join the concentration. I teach one of the first classes in the program, an introduction to genomics, and try to put that into context. So with that, I think we just let any of the students have their questions if they have any.
Thanks, Joe. I wanted to add one more note to that. So I come from a programming background. And so, as I've mentioned several times now, data is data. So you're going to be learning genomics data, genetic data, but you're also going to be learning programming, and you're going to be learning working with large data sets and data management and all these things that, should you find yourself going in a different direction after the program ends in a different discipline, these are all going to be applicable to whatever you're going to be doing. If you're going to be staying in the sciences or even going into business, I mean, everything is data driven now. So I don't want you to think that you get this degree and you have to go into genomics. I mean, there's huge opportunities in genomics, of course, but there's huge opportunities in data itself. So I come from a little bit different perspective. You know, I never thought I would actually become a professor. I wanted to be programmer forever, but then I found a different direction, and here I am. So it happens all the time. People do something and they, you know, change directions right in the middle and find yourself somewhere else. So this program is extremely relevant to you know, any kind of job you're going to be going into after college. So.
Yes, and we know that your background, you're not a geneticist, but you
Right.
handle big data sets in GIS, right? So
Yep.
that's related to biogeography and metadata that's necessary to actually put some of these sequences into context.
Yep, and I also do data management. I mean, when I was a programmer, I actually wrote database systems, and I, in my own research at Nova here, I've used massive databases to run biophysical simulations. So, you know, I've been able to do all these things because I had that foundation of programming and data management. So again, these are skills you're going to be taught. So they're very applicable to whatever direction you want to go in the future.
So I had a quick question for Yvonne. Just I know you can't tell us any trade secrets, but with regard to, you know, the biotechnology field, do you see opportunities there? I think you mentioned that in your just description, but anything else on the horizon that's kind of really?
Well, you know, biotech is an amazing but very competitive world, I would say. You see biotech flourish all the time, and there is opportunities for everything. That's why us in the lab, whatever you're doing in the lab, is great, because you will use products from multiple companies that will be created every single day. And I mean, biotech is why it's how we progress, right? How we discover new things, because people are constantly innovating and that kind of thing. As for us right now at Jumpcode, we're rather limited in terms of space, because unfortunately the biotech space, as competitive as it is, and I think everybody right now knows about the climate that we're in, like economical climate. So biotechs are trying to pull back a little bit, but the bioinformatics side is not only applicable to biotechs, basically. So you can do bioinformatics without being part of a biotech. And that's where the whole power of the program and those skills are, because you can be applying them no matter what you're doing, basically. But yeah, no, the opportunities in biotech are always available, and it's just a matter of trying to find exactly what you want to focus on. Obviously nowadays, you know, a lot of the interest is based on cancer therapy and human comfortness, I would say. So there's a bunch of biotechs and companies that are developing products and needs analysis done to interpret the data they're generating based on a new drug or based on a new product that helps them detect cancer earlier, or that kind of thing. So definitely those skills and tools are necessary to analyze that kind of data.
Excellent. Yeah, in regard to human work, I mean, the one example is CRISPRs. You know, you really can't do a lot with CRISPR technology, but that's kind of the big thing these days. But you need sequence to work with CRISPRs.
Yeah, CRISPR on top of that requires like what we call sequencing depth. And for those that are not familiar, is how often you sequence a single molecule in order to be able to make sure that whatever you're targeting is the actual sequence. CRISPR is sensitive enough that it can recognize a single nucleotide difference. And so if you have in the DNA strand, for example, if you have a what we call the SNP, CRISPR might not be able to insert a sequence or cut that sequence. And so that's where biotech and bioengineering really comes into play, because if you do not know these data and these aspects, then most of the technology are not applicable. So, but back on CRISPR, yeah, our company uses CRISPR to clean up sequencing libraries and basically remove background noise, which in turn reduces sequencing costs. For example, we have some projects that are looking at differentiating cancer-related genes and remove the wild-type genes to see whatever the gene is, the new gene responsible for cancer is. So you would sequence data, you would sequence your DNA, but that DNA is depleted of the wild-type gene, and so that enhances the detection of possible cancers. So this is, and then, but obviously, while you do that in the lab, you cannot know for sure if it worked or not after you analyze the data. So you sequence, you get the data, and you analyze, and you see the difference levels in the wild type versus the allele specific gene. So again, bioinformatics come into play in that case, because you would need something comparisons between your non-treated samples versus your treated sample and that sort of stuff. But obviously, I cannot divulge all the information because it's all protected by NDAs, but just to give you a quick snippet of what can be done.
So
Yeah.
Yvonne, in a general sense, you know, one of the technologies that has created tons of buzz in the last year or two is artificial intelligence, which we'll talk about that a little bit in the biological data science course about what it is and what it is not. Are you seeing AI making any inroads as far as your work yet? Or is there
that's a very good point. AI is a hot topic right now. You see a bunch of new AIs popping up, being like, I think there's a new AI that just popped up and that is able to predict very early on with a blood sample sequence the prevalence of a cancer in 75% or 78% of the cases, which when you think about it is phenomenal, because you can be aware that you have an issue early on and then be able to get treated. As far as the biotech world, I think the AI will probably be leveraged more so in automation and pipelining everything, in sense of you just ask the AI, okay, I have this data sets, that's what I want to see. And the AI will be able to fetch the different softwares that are required to analyze the data, and basically will do everything on its own. And you won't even need to have like an AWS instance or a server or anything. You just tell the AI, okay, here's my data sets, here's what I want to see, do your thing. And so in a world where computing is just becoming huge, the AI is certainly going to help us, and in my opinion at least, fasten the processing of the data and also the interpretability. But the big drawback is that obviously the AI is a system that is being trained over time, right? And so the more data sets you're going to do, the better it's going to get at interpreting the data. The drawback is that you're still going to have to go back and make sure that there's something wrong that didn't happen, right? So it's a give and take. I see it as very helpful, but there's probably going to be times where it's going to tell you a result and you're going to be like, I'm not really sure. You're going to go back and you're going to see that the software that was used, the right settings or the right parameters are not being used. And so it's a little tricky, right? But long story short, I think it's a great tool, and that will definitely be helpful once we refine the algorithms and the way it works.
Yeah, AI is kind of somewhat in its infancy, and it can, I talk about it in our course, it can be kind of a slippery slope, because it is trained on data, but if the data it's trained on is wrong, then it makes wrong decisions and can come up with all kinds of, problems. So there needs to be a lot of checks and balances, but that's one thing you'll learn, in one of your initial courses that you take. So I was curious about if it fit into your line of work yet. I'm sure it will.
Unfortunately, we haven't really experienced it because a lot of our pipelines are already written down, and you just grab the data, push it through, and we're done. We're getting out the data, or the figures more so. But we've worked with a company that does AI to interpret meta transcriptomic analysis, and they're not far-fetched of what we find. But obviously, the AI doesn't have the granularity that a human can go to, especially with some softwares. So it's and again, right, even if you get the perfect AI that will work and give you all the data, then it's up to interpretation, right? So you have to really go in and understand the biology behind it and really make sense of what the AI gives you. So it's certainly going to help some part of the process. To the full extent, I have my limitations on it for now.
Yeah. I've heard that it actually could be useful maybe for code. And I'm not a coder, but for
There's going to be a
scripting.
big shift in the way
So-
things get programmed, and the tools that are developed to create programs. Not that programming is going to go away, but the way it's done is probably going to change quite a bit. So instead of handwriting lines of code, you may be relying on an AI thing to write things, but you're still going to have to check what it's doing and still know how it all works. So there's just kind of going to be a big shift, not away from programming, just using different tools to do the program.
I guess the tech devs and the web devs are a little bit of a problem ahead of them.
Yeah.
Yeah,
On to
definitely.
the-
Like anything else, you have to be careful.
I was going to add on to the discussion on AI. Those are excellent topics, and how the AI is used in automation and coding and making pipelines. And we have a course for machine learning in biology, which is one of the courses in the last semester that you will take, where we will focus our shift on how we can use AI to make genotype to phenotype predictions. So given the amount of data that we have available, public, free data, we are going to ask a specific research question, and then we are going to train the data set based on what we know, all the possible information we know about the data, create a very comprehensive data set, and then use the data set to generate AI models, machine learning models, and then make phenotypic predictions. And that goes to the point discussed earlier in how those predictions early on can be made for any type of disease or something that's unforeseen for the near future. So that will be a cool course to take, and doesn't matter what you're going into. If you are a computational biologist, or if you're going to biology, biotechnology, courses like that, as well as metagenomics taught by Dr. Ozga, and transcriptomics, for example. I mean, if you are a medical researcher, you're working on cancer biology, you need transcriptomics to make sure you understand the differences between the genes that are expressed at a higher or a lower level in diseased versus healthy tissues. So it doesn't matter if you're a pathologist working. I see a question in the chat where they asked, like, are there any research opportunities under this program to branch to microbial pathogenesis or experimental pathology? Yes, all these courses you can target to whatever field you want to go into. Like Dr. Lopez mentioned that, and Dr. Johnston mentioned, data is data, and every cell has their own set of data. It's a very system agnostic. That's my favorite phase for bioinformatics. It's a very system agnostic field of study. You can use it in any system on planet, and you can use it for every field these days, because there's data all around us. So this is something that is very dynamic field of study. If you want to go into computer sciences, you have the courses, you can pick the electives that you can use in that. If you further want to study more into computers, if you want to go into community studies, metagenomics, you have that knowledge, you have the basics, and then you can add onto those. cancer research, medical biology, any field of study that you want to go into in future, you can rely on this course, I mean, to give you the basics that you will need in this field. So, yeah.
I wanted to just add one quick thing. Yeah, I totally agree with your assessment there, Dr. Gill. And you know, the way you know, at least me in particular, teaching courses in this field has been really fun, because I do change the course every time, and I also mold the course based on what the students want. So with the metagenomics course, there's a project that involves you seeking out a few papers and some techniques that you would like to get better at that maybe are brand new. That's the whole point of this, is that you're understanding the foundational components of this data science and of these biological, you know, huge biological data sets that, you know, we can together look at this paper that just came out last week, and they have an open code that's on GitHub, and we can start using some data in replicating what they're doing there. So I think that's really the perk of this stuff, is that we all want to you know, kind of challenge ourselves and try out some of these new things, and we're not really limited with how we, you know, what we can work with. If someone comes to us with a particular idea or paper or data type that we don't know about, you know, chances are somebody here or someone that we know, you know, colleagues can kind of walk us through some of the challenges with that stuff. So I do really like that. Yeah, the dynamic and flexibility that this track has within our master's program here.
And that appreciate Tiffany asking that question, you know, on microbiology. She was one of my microbiology students, so that's great. And then Dr. Ozga is going to teach a metagenomics course, which focuses on a lot on microorganisms, and those are wide open as well. I mean, we are, you know, just at the tip of the iceberg of trying to understand how microbes really direct and are important for our planet's survival and what they do for various ecosystems. So, you know, microbes have much smaller genomes. We get to access them easier, faster, but they do a lot. And we want to understand how they work in symbiotic systems, free living systems, and we can ask those questions. And there might be, I've heard from other students, not here, but doing some work on the ground or here, and that'll be easier if you're in town, and we could possibly, you know, work things out that when we have you have a laboratory question, we might be able to bring that in. So, you know, that's something that could be adaptable. But overall, you know, this was meant to be an online program to be convenient to students who aren't on campus or nearby. And so we want to keep that aspect as well. And because this is very computationally based, this allows us to do that, you know, we can have that flexibility, and you can learn all you need to know from the experts and the faculty here to carry out using these big data sets to learn those skills on various aspects. But again, we can offer people who are in town or locally to you know, come visit, take the class, maybe as a hybrid class. I teach genomics as a hybrid, but also visit our laboratories and work with our faculty, maybe on a project that they want to see one-on-one, or if they have a data set that once they've taught you those skills, you might be able to take that data set even further, you know. So again, I think we see a lot of opportunities and flexibility here with this program, because we're just starting it out, and it's exciting. Exciting to be.
And also an added advantage or incentive for folks who want to attend this on ground is that the department is offering to pay a stipend of about $5,000 per semester, and a TA-ship is also available for students up to $5,000 per semester. So that kind of makes it to about $30,000 in financial aid, so to speak, over the term of the whole course. 15,000 in TA-ship and 15,000 in stipend. So that is like if you're local or if you're around, so that would be a good incentive to kind of take care of the costs involved in the tuition and other fees. So that would be a great help, something to think about if you can be on ground.
Thank you all. If we don't have any additional questions, feel free to reach out to me. My name is Megan Troy again, and my email is mtroy, Troy, @nova.edu. I am the guide to the admissions to get into this program. So if you have questions regarding admissions, please feel free. And if you have questions for any of the faculty, I'm sure they are all more than willing to answer an email or two. If you have questions, you can feel free to reach out to me and I'll get you connected with them. This is recorded, so I will be publishing this on our YouTube, our Halmos College YouTube page, probably early next week once we have it edited. And then I'll send it out to all the participants that were unable to join us today. But again, thank you so much. If there aren't any further questions, we will go ahead and close the meeting.
Thanks, everybody, for your participation.
Thank you all.
Thank you.
Thanks, folks.
Enjoy the rest of your afternoon.